back to school days. Too much homework.
So welcome to Chulalongkorn University. Exactly.
Yes, exactly. But actually, it's good.
It's good, nah. Yeah, shall we
Yep.
start right now? You all ready? Okay, if you're ready. So today, yeah, we gonna talk about the microbiomes, but just only microbiome, maybe not applicable to any side of for our user. So right now, I think I'm gonna convert some content to integrate with the how to use under the applications of the probiotics, prebiotic, and postbiotic. So let me share my slide. So actually, this slide has been used yesterday for Hughes seminar. Yeah, they assigned me to talk about the miracle of gut microbiome, especially for cat. But I guess this may be useful for this class because, yeah, on the same times we're gonna talk about the gut microbiome. Okay, to make sure you all got the handout. You got a handout from the new conversations I just posted in the work team. The team. Please go to the first page of the Microsoft team and you can download.
Okay, Ajahn.
Okay. So you all have pet in your house?
Not really, Ajahn.
Not really?
No.
I have one rabbit, one cat,ค่ะ.
You got one rabbit. And shall we?
And cat,ค่ะ.
One cat.
I don't have.
You don't have, but you still wet, right? You have to, yeah, give this information is to all the owners. And so I think that there is a lot of things that can contribute to the life, to your life, your animal life, that can make you are what you are right now. But importantly, one of the things that can build you to be like this, especially feed or food. So what the feed or food can make you as you are today. So today we're going to talk about the how importance of the very important organ that can contribute a lot of chemical substance to heart, brain, liver, bone, blood vessels, all the things in your bodies that originated from gut. And also what inside of the gut, we're gonna focus on the intestinal microbiota. And how can we enhance the integrity of the gut by using of the probiotics, prebiotics. And we need to know about the indications of the postbiotics after productions of the some chemical compound from probiotics in gut and how it's bring to the target cell like neuron cells or adipose cell. And finally, it's going to spread to the safety and qualities of this kind of the products, how to use, how to make the considerations criteria. So let's go to the first page. So you see this life comprising is the root, the nutrient. Is my point up here? Okay, so this is life. It's comprising from environmental part, their own DNA part, and also the nutrient that feed it from root. So therefore, this one, oh my God, yeah, appear from in several parts. Firstly, we can observe the appearance of the life. That's the way you are right now. So you're still healthy. Some get sick, some get diabetes mellitus, or some get allergy. This is the appearance that happen after you living like your own lifestyle. But actually, this is made from the metabolic imbalance of your life. The problem from these core metabolisms that can bring all the system to inflammation phenomenal, stress response, or some others in ordering phenomenals. But actually, this is from the root cause. The root cause comprising genes or epigenomic or environment and the nutrients. So all the things comprise to be one life. That's why I ask you to pay attention on the most important thing is feed. Because of the feed can contribute a very useful or benefit microbiomes that can produce some miracles chemicals to via the gut, and this kind of the chemicals can transfer to a lot of organ in your bodies and makes happy life. And then look at the gut integrities and what is it and what we expected from the gut integrities and the immune regulation. So look at these ones that can describe very well about the usefulness of the gut, that once they can work well, they can synthesize protein, or they can regulate the immunities, and they can contribute the liver health, and also basically they can synthesize the enzyme like properly. And this one maybe not so familiar because they can synthesize the neurotransmitters and hormone also. This is a lot of things can control overall of your bodies. That's why the gut function is not just only digestions and pass all the nutrients, but these parts can bring life, homeostasis, controlling behaviors, emotion, and the performance. Before you will understand about how they can integrate by functions, but we need to know, so how do we pay attention on the immune system, especially on the mucosal immune system, because this part is very huge part, the wide, very wide. Maybe your mucosa will range at least 400 square meters. Let's see, this is equal to futsal field. It's very big, and that represents how hard work that the body need to protect the this kind of mucosas against to all pathogens or chemicals not too harmful to your bodies. That's why this part is very important and this part is directly concerned with the nutrients and microbiome. And also by functions, their work for balancing and controlling of their own immune protections and immune balance both ways. They have to secrete some mucus or antimicrobial peptide from the cells to outside to against, and also they have to produce immunoglobulin A, and also some physical barriers to protect the bodies from all pathogens. And this regulation is very important because if the immune system got confusing about pathogen response or non-pathogen response, or even the response to their own cell, they're gonna destroy their own cells by themselves, or if they get overreactions to the pathogens, of course the normal functions will get worse or errors and cannot adjust to the normal conditions. That's why mucosal immune system is very important to control overall of the bodies. So let's see what is the immune systems behind the host cells, behind the epithelium cells. Under the epithelium cells, so you can observe a lot of effector cells such as neutrophils, so you're familiar with this one, eosinophils, macrophage. And this one work like normal soils that can kill all the bacteria or some pathogens like yeast cells, some of the yeast cells. But anyways, their own functions, they have their own very important function is to present the antigenicities to some other cells, and that other cells become the spatial soils to decide what the bodies will have which direction. That is the helper cell. So one, the antigen presenting cell like dendritic cells or macrophage, they got the foreign cells and they presented to the group of the helper cells. And the importance predominance helper cells comprising T helper 1 and T helper 2. And these two of the cells have to living in balance, not over influenced by some of the cell. That's why balancing of immunity is very important. So how they can balance the cells? So they need to have like commanders to balance the cell. This kind of cell become commanders we call regulatory T cell. This is a FOXP3 positive T cell. So these mechanisms or reactions can work well under some commitment. And the important commitment would be microbe. Because the microbe, if all the community of the microbes have their own like good population and bad population in balance, and also if they can if we can observe the diversities of the microbe, yeah, of course that microbe will be that microbe group will be very useful for the body because they can regulate each others, and they can induce some new neurotransmitters, and also they give some like a good practice from gut to others organ. If the gut can get practiced well, some the others organ like liver, kidneys, brain, bone marrow, or even heart will get very healthy as well. So how they can work? This is the pictures represent the interactions between pathogens outside at the lumen. This is a mucosa, and this is a lamina propria and under the mucosa. The effector cell and T cell, the helper cells, build linings under the mucosa and ready to learn what kind of the pathogens that's gonna approach inside. And the important main white blood cell is neutrophil. This is the common cells that against to bacteria and fungus. And after they learned, oh, this is bacteria, they usually secreted the cytokine, especially the IL-17. And also they can trigger the inflammatory cytokine together, especially TNF alpha. And some of the part of the bacteria can directly introduce themselves to macrophage. And this macrophage not stop like by killing of the bacteria, but they can bring some of the part of the bacteria to some other helper cells, especially T helper 1. This is the messenger protein we call cytokine, right? Yeah, the cytokine, the important cytokine that reflects the pro-inflammatory cytokine phenomena is the TNF. And for interferons, this is the cytokine for recruiting some of the T cells or some other killing cells to the infective site. This is very important. So that's why this can work well like a team. But anyways, if they can recognize some helminth, yeah, of course, the way of communication is changed, will be changed, and it's changed to T helper 2. So after they recognize it by some of the cells can differentiate some other plasma cell to be like T helper 2 cell. And the T helper 2 cells will differentiate another plasma cell and convert it to the mast cell. The mast cell is ready to be degranulate the histamine. But of course, this pathway need secreted of the IgE instead of IgG or IgA. So that's why this is very severe once the body is designed to go to these directions, and these directions will hurt mucosal immunities directly. Or so we can notice what happens after this. Of course, is it diarrhea or is it asthma? This is the response after T helper 2 response. That's why the body don't need this kind of the thing. Let's see how to control the T helper 2 and switch to T helper 1, or not to secrete too much pro-inflammatory cytokine. So let's see, this one is the group of the beneficial bacteria that can trigger some of the miracle cell. That miracle cell is regulatory T cell. And this kind of the T cell will inhibit the over differentiation or the recruitment of the T cell or inflammatory cell. So that's why the beneficial microbe can protect the bodies from overreactions, especially in terms of the allergies. Then let's see what's going on. So if we know about the health benefit of the bacteria, that's why bacteria can control all the things in your body that toward bacteria could make a big change in the host. So from the gut can control brain. So like this affects is the sensitive dependence on the initial conditions, which is small change is one of the stage of the determining non-linear system can result in the large difference in a later stage. So from these pictures, we can guess this is the modern pathway that they can explain how relations between brain and gut, because we have this word gut-brain axis. That means the gut can influence on neurotransmitter stress, and anxiety and mood and behavior, even your behaviors. Gut can control that. Amazingly, the brain also can influence to the gut, especially in terms of the mortalities, secretions, nutrient, and microbial balance. So the brain to gut, like the top-down policy of the company is that, so we need to understand, once you get stressed, you listen to my lecture and you get very stressed, so why I cannot communicate to you well and I don't understand what you say, your English is very poor. You get stressed, right? You get stress induced and your neurotransmitter released. And then, of course, this is the neurotransmitters makes imbalance of the T helper cells by the neurotransmitter got catecholamines, dopamine, and serotonin, and a lot of the hormones that involve the cell communication. So once they get imbalance of the T cells, they can suppress the T helper 1 and promote T helper 2. Oh, this one is very evil. So how bad of the T helper 2, the evil one, that is the secreted of the mast cell, and they can degranulate a lot of that the pro-inflammatory cytokine and also prostaglandins or some unwanted chemicals that induces the heat of bodies or fevers or something, and they move to the gut dysfunctions by increasing of the eosinophil, as I said, and weaken the gut barriers. This is the pathway of the gut-brain axis. So come to the unwanted conditions that we call, if the imbalance happen in gut, we call dysbiosis. Then this is the abnormals of the microbial structures which affect the taxonomical compositions as well as the metagenomics function. So this comprising three type of the dysbiosis. First is the bloom of the pathobionts. Patho is bad thing. Pathobiont, the bad bacteria, and loss of the commensals, loss of the diversities. Excuse me, do you already recorded, right? Shavin?
ครับ yes
ครับผมจะได้ใช้ในปีหน้าครับ จะได้ไม่ต้องพูดใหม่ ขอบคุณครับ
ครับ
Okay, look at the picture. This represents the overview of the dysbiosis conditions. So in the normal conditions, we call immunological equilibriums. You can see the equally populations number of the symbiont is the beneficial bacteria, the commensals and pathobiont. This is work together and work well in terms of the regulation, and not that too much inflammation, that this response just enough to protect host membrane, and the regulations to control this one by symbiont. On the other hand, if we can see immunological disequilibriums, that means some of the part get over or predominance. So this part represents the overpopulations of the pathogens that can affect to over reactions by inflammations that against or that impact to the host, especially on the gut balance. So what can make this biosis in your life? Host genetics, of course. Some of the populations get very easy to get worse on their own stomach, but importantly, we got commonly effects from lifestyle. So you drinking alcohol, and you eating a lot of Western food, You get stressed every single second surrounding you. Everyone can upset you. So I want you. So we cannot control some other people to get upset, or you actually you can control yourself. You can program yourself that today you're not gonna be what to say, be called proactive. If you program yourself to work today, not to be slave of some others emotion, then you can grow by your own, and you can work by your own idea, your own emotion. Then you don't have to care about some others stress that hit to your life. Then right now you can control it. Believe me, you need to get proactive in your life. And also, if the medical application is working on you, like antibiotics and vaccination use, or even too much hygienics, this one affects and are being the cause of the dysbiosis in your life. And what happens after you get this imbalanced condition? Of course, you will get the disease under the overpopulations of some T helper cells, especially T helper 2, but some T helper 1. Of course, inflammatory cytokine that's secreted from 17, T helper 17. And if you want to convert these conditions to healthy, you need to consider importantly on the T regulatory T cell. And then if you get this biosis, so what will happening to your gut? Let's see the right part. So once the beneficial bacteria get lower in the numbers than those of the harmful opportunistic bacteria, the gut permeabilities will be increasing together with increase of the enterotoxin. And they have the potential to be like bacterial translocation, that the gut can, the bacteria can move from lumen, from mucosas to the blood circulation. And also the systemic inflammations will very increasing. And some of the hosts get high level of sugar. That means that patients will get in conditions of insulin resistance. And this affects to the adiposities, DMs, or some other endocrine disease. In contrast, if the balance of the gut microbiota happen, that means the beneficial bacteria and commensal will get over or get influence over the harmful opportunistic bacteria. This can convert these conditions to be the opposite side. Then the patients will get recovers, get healthy, low risk of infections, and can control the blood glucose. So what is the beneficial bacteria? Yeah, we're gonna focus on the bifidobacteriums and lactobacillus, or even Staphylococcus. We call all of them is lactic acid bacteria. Then look at the bird eye view from gut, from about the benefits and dark side and bright side of the gut. Of course, the gut have their own functions. They have to work well to balance and to digest, give the nutrients to bodies. But at the same time, if they got imbalance, they will be the cause of allergies, weight gain, asthma, autoimmune disease, and some of skin disease, even cancer. So look, approach to the intestinal microbiota. So you can see the this kind of the number that related to the microbiotas. So actually the microbe will be consists of bacteria, fungus, archaea, yeast. This one, the most populations numbers will come to bacteria. 98% of the gut microbiome is bacteria. That's why most of microbiotas will report about the bacterial component, not that much viral component or yeast component, because 98% of the populations is bacteria. And in terms of the numbers of the variation species, they have over 500 species that comprising in the gut microbiomes. And if we can get 1 gram of the feces, we can observe quite a lot of the bacteria inside is maybe reach to 10 to 14 is equals to 100,000 CFU. So that's why we never be alone. We have a lot of microbiome inside. And yeah, of course, if you're really lonely, you can talk to your microbe. And if you accumulate all of the microbe and we weight the microbe in your bodies, you will get the microbe equals to 2 kilogram. That's equal to liver weight. That's why we call the microbiome is one of the important organ that's necessarily equal to liver. So where is the microbe? So if you observe along the GI tract, so you can see from the mouth, right? Mouth from stomach and come down to small intestine and to colons. The conditions of the acids will be decreasing from metabolisms of the bacteria, and the bacteria will be increased until the colon. So you can observe the bacteria from 10 to 10 or 10 to 11 or 12 at the colon, not at the stomach. That's why the alkali conditions can enhance the populations of the bacteria. And this one will respond all of the things around you and will direct your life. So why we have to understand and study about the microbiomes? Sorry, I'm writing in Thai. So because we think about the gut microbiomes or the microbial environment is like one of the socials in one area. If the socials comprising the diversity of the bacteria and that bacteria can accept each other to live together, that would be nice. And if that bacteria or that human populations know their own function, know the responsible to their duties, and they have their own abilities to contribute to the social, of course the socials will get very strength and strong. And if they got some change in gut or social get like we call change of the socials, this disruption, social disruption, the peoples can adapt themselves and understand how they change, and they can live together as well. Of course, that kind of episode will just bring growth of the living. So the bacteria as well, if they can facing with some infection some antibiotic, some allergy, and they can pass that one. Of course, they need to adapt themselves in terms of the populations, and some of the bacteria will become dominant to against that some disease condition. So finally, all of the bacteria, all of the 500 species have to live together, right? So if they live together, they need to have just one ultimate goal. So the ultimate goal of the bacteria is just for living, just for their ancestor to contribute their host. If host die, of course, all the bacteria die. If the social collapse, if the nations collapse, all the people have to be rely on some other country. So we don't need that. So that's why the microbiome is very important part in the bodies and their life, and what the microbiome work in terms of the digestion. So let's see, this is the normal term of the food, the feed go to the GI tract and along to the intestinal gut until go to the feces. So once pet eat feed, the loop of the digestion will be divided into parts. The first part is the host digestion. The second part is the microbial digestion. So this is the normal term because the in feed will comprising the carbohydrate, protein, and lipid. And the host digestions will work very well in terms of the enzyme. The enzyme can digest this kind of the nutrients to be a smaller molecule like mono or disaccharide, amino acids, and fatty acid. And the host will ready to absorb this kind of the nutrients and yep, of course, feed to some others organ. In contrast, the microbial digestions will also work very well with the non-digestible carbohydrate, and they can contribute to produce the oligosaccharide and some of the short-chain fatty acids and some of the amino acids, and of course including the vitamins, folate, and some others mineral compounds that is necessary for support as a nutrient to the host. So that's why what we need to pay attention is some of the veterinary who work in feed or diet, they need to calculate the proper formulas that can deliver the nutrients and enough to pass these host digestions and still have some leftovers to the microbial digestions until they get the postbiotics to support the feed to the bodies of the host. So what difference between hosts? Yeah, this is the one of the basic that the people who work on the microbiome need to know about the requirement, the basic requirements difference in each species. For examples, the cat we call obligate carnivorous. The obligate carnivorous need very high protein diet compared to the carnivore like dog. But of course, we know about taurine is very important for cat, but not just only this. One, they need to produce some a cat formulations feed. They need to think about the high protein diet, and they need to digest some of the carbohydrates to be a smaller amount of the carbohydrates. Otherwise, the digestion of the cat will cannot digest well on the big pieces of the carbohydrate. By bacterial diversities, you can see this is the number is difference between cat and dog. The cat has much higher taxa of the microbiome than dog, and comprising a lot of member of the yeast or fungus that the dog does only have Nagasomyces, but the cat has some others yeast or fungus species. So bring to the overall of the lectures about how this can make work well together from the beginning until the end. Yeah, from the beginnings. So we think about the nutrients or feed. And recently there is a lot of commercial product that can appear in the advertisement about the prebiotics. Some of you ever bought prebiotic that they claim once they take prebiotics, so your GI tracts will be healthy, and it's very good in the morning if you come to the toilet. Yeah, we're gonna split the topics as prebiotics and go to probiotics, because the prebiotics can support and promote some of the microbe called probiotic. And the probiotics can produce some of the miracles chemicals we call postbiotic. And the outcome from feed and balancing of the microbe will get the very excellent outcome of the health and fecal condition and some of the treatment for especially allergy. So by definitions of WHO, prebiotic is a non-digestible ingredient that's added specially into diet and use that component to contribute or modulate the growth or metabolic functions of the resident bacteria. Go to the probiotic. This probiotic is the live microorganisms that, when administered in adequate amount, confer a health benefits on the host. If harmful to the host, not called probiotic. And this is the final product. Is some of the companies, they produce the symbiotics. That symbiotics mean the combinations of the prebiotic and probiotic together in one product. Then firstly, we need to know about how importance of the feed diet that can get dangerous into your gut. So from the nature's journals, they recommend importantly about Western food. Yeah, they said about 75 of all of the food in the Western diet is of limited or non-benefits to the microbiota in the lower gut. But most of it comprising especially on refined carbohydrate. Yeah, this carbohydrate is ready to be absorbed in the upper part, like small intestine or jejunums of the gut. Therefore, there is a limitations not left enough to a large intestine. So that's why the protein or minerals or some important nutrients cannot be maintained in this part and cannot contribute some other beneficial bacteria at the colon. So what we need to add up in the feed. So some recently from the scientific research, they recommend prebiotics because the prebiotics will contain, the important thing is the fiber salt. For example, like FOS stand for fructooligosaccharides. the acacia or gum arabic, pectin, inulin, the very indigestible starch, or even beta glucan that is a part of the yeast. So this kind of the nutrients can like can promote or enhance the bacteria to produce some of the miracles components like short chain fatty acids comprising acetate, propionate, and butyrate, and some of the antibacterial activities, anti-inflammations, and antioxidants. This will synthesize via the carbohydrate fermentations pathway. So let's see the kind of the carbohydrate. You can see the right and the left parts that represent the soluble fibers and insoluble fibers. The soluble fibers is mean if some of the carbohydrate can solubles, of course they can be metabolized by microbe or bacteria, and they can the bacteria can convert some of the nutrient inside or some of the chemical inside of the soluble fibers to be benefits to their bodies, and the benefits will be like stay full of larger, so you will feel empty. Do not feel empty, you feel full longer, not need to take that much feed. So they can this kind of the feed food, this kind of the food can prolong hungry time of yourself, can diet can help you in terms of the obesity. Yes, and they can decreasing of the blood cholesterol and blood glucose. This one. On the other hand, this one is insoluble fibers. The insoluble fibers, if you ever buy some of the product of the fibers, and yeah, you use the powder of the fibers and pour to your glass, and you pour the water inside and let it set in like a few minutes. You can see the volume of the solutions will get high, a little bit higher, and the texture of the solution is not water anymore. It's gonna be like gelatin. So this is insoluble fibers, and insoluble fibers can make the stool conditions is proper, or look the fecal score get better and can prevent constipation. And what other else inside of the food, this kind of the carbohydrate? Yeah, the important chemicals we call polyphenols. What is polyphenol? Is the phytochemicals that originated from vegetables and fruit. So you can see the phenol rings here. If there are over 3 or equal to 3 phenol ring, we call polyphenols. And this polyphenol will be converted in the stomach, in the colons by microbiome, and become a miracles of antioxidants chemicals. And also in the gut, in the plant, vegetables or fruit also contain 1 or 2 rings, not a polyphenol rings, but we still call it, this is a phenolic compound. This phenolic compound also act like anti-inflammations and antioxidants. So you may familiar with resveratrol. It's a very miracles antioxidant, astaxanthin, right, or something. this one is antioxidants chemicals that created from plant, and they use that kind of the plant and the mixture of the polyphenol in the plants that for treatment of some of the disease, especially for protecting the cancers, the neurodegeneration disorders, or thyroid disease, the asthma condition, liver disease. Quite a lot of disease can be recovered from polyphenols. So let's eat fruit and plant or prebiotic. So what we get the benefit for the polyphenols, that including flavonoid and non-flavonoid. Yeah, this can down regulate the fevers chemicals, prostaglandin leukotriene, and can down regulation of the angiogenesis. This one is a mechanism to build up a new red cells to carry or to maintain the cancer cell. So then they can reduce the blood vessels go to cancers, and also get balancing of the pro and inflammatory cytokine, anti-inflammatory cytokine together. Then the sign of the allergies will get recovered, and also they can reduce the regulations of the oxidative stress. And recently, they have the evidence to upregulate the autophagy. So anyone here know about the autophagy theory? Silent? Okay. Yeah,
No.
the autophagy is the mechanism of the cell that can eat themselves and eat their own self. It's a mechanism for recovering by natures that happen in the cell level. This can enhance the lifelong age and reduce the opportunity of the some of the disease appearance. Actually, this theory has been crowned by Nobel Prize by Professor Yoshinori Osumi in 2016. Yeah, he discovers this mechanism, and they can understand how to deal with Parkinson's disease and how to reduce the cancer or like an anti-aging mechanism. So there are 4 parameters that they're gonna observe after they give the prebiotics into the host. Firstly, so they can contribute on the feed intake and can improve the because stool parameters and can balance of the microbiomes and can enhance the productions of the short-chain fatty acids called postbiotic. So the feed intake and change weight can see in the obesities patients, and also once the scientific articles reveals about the benefit of the prebiotics to against obesities, chronic colitis, and allergies. This is one of the success models of the prebiotic. And also they observe just 2 parameters that convert the fecal grade 1, 2, and 3 to be 4 and 5 successfully. And also in terms of the microbiomes, they can enhance the bacteria that can digest well the carbohydrate, we call saccharomyces bacteria. And these bacteria comprising, for example, the Clostridiums, Bacillus, Aeromonas, Pseudomonas, and this will be enhance the population, some of the populations, especially Ellanospiraceae bacteria. This is studied by microbiome analysis, and this can reduce the bacteria that work well on the digestion of the protein. But some of the bacteria can work well together, like can digest both protein and carbohydrate. But the pathway of the protein will be inhibit some of the bacteria. What is the waste product of the protein digestion? Amine, right? New ammonia, urea, they are toxic. So we don't need that one. So we need some of the bacteria that can reduce that one to destroy the byproduct of the protein. For example, like the sulfur BPO species. This one can we call use the nitrogens in the gut intestine. The last part, the scientists go to observe the probiotic. Both of the probiotics, this one, short chain fatty acids, in the formula of the strength change and branch change. And what difference? The strength change produces from saccharolytic bacteria, but the branch change that produces from proteolytic bacteria. So this one should be better, right? Because this is from proteolytic bacteria. Of course, the outcome will be acetate, propionate, and butyrate. And this will directly affect to benefits in terms of the anti-inflammatory effects and reduce of the pH. So this can modify the acid condition. It's very good condition. But on the other hand, so if the mechanisms go to enhancing of the probiotics, so enhancing of the proteolytic bacteria, these kind of the bacteria can produce not strength change short chain fatty acids, but will be branched chain short chain fatty acids. The branched chain is the toxic compound, and they also produces the branched chains of amino acids like valine, isoleucine, and leucine. This is a toxic for the cell. Then what we can make a correlations between the probiotic and prebiotic. Yeah, of course, look at this kind of study. So they feed the prebiotic like phos and cellulose. Cellulose is non-soluble. Or the phos and pectin. Pectin is soluble carbohydrate. Two of these formulas can induce the beneficial bacteria like bifidobacteriums or lactobacillus, and can decreasing of the E. coli or Clostridium perfringens. So from this kind of the studies can confirm the relativeness between prebiotics that can enhance the beneficial bacteria. This brings to the understanding of the probiotics, because the probiotics have their own functions in the gut. Of course, they produce several antimicrobial compounds, and they can like subsidize the communications between the cell to cell in the bodies. So then probiotics like friends. If you got friends, yeah, people have friends. What kind of the friend you want? The friend who give you money, the friend who give you nutrient, the friend can convert some bad communications to good communication. the friends who bring warm batters, the friend who subsidize all of you, yeah, that is probiotic. This is all of the benefits of your friends. Then what we expected from probiotics. So this is the indication: antitoxin, anti-sporulation bacteria, anti-biofilm, reducing plasmid transfer, and antibacterial activities from antimicrobial peptide, antifungals, anti-cancers, or reducing of mutant inside, especially cancer. So anyway, the releasing of the product of the probiotics has been controlled by international formulation regulation we call GRAS, generally recognized as safe by the FDA. The FDA will have their own regulations, especially the in terms of the characteristic of the probiotics that need to tolerance to acid and bile tolerance, not to produce too much phenols, and not contain the genetics like antimicrobial resistant gene that can transfer to some other bacteria. Then they have to investigate whether they can produce some or contain some of the resistant gene that can transfer by plasmid, for example, to ampicillin, chloramphenicol, macrolide group, and vancomycin, tetracycline, and streptomycin. Yeah, of course, between dog and cat, initially I have presented one slide that the community of the bacteria is different, and the member of the beneficial bacteria of dog and cat also different. So this is the member of the beneficial bacteria, especially for lactobacillus, and this is for lactobacillus in cat. Some share species, some not share species between dog and cat. But anyways, even consider just only species is not enough to produce as the probiotic. We need to think about the strain specifics. Therefore, the lactobacillus acidophilus from one dog cannot use for cat, or from one dog without any characteristic cannot use as the bacteriocide to be produced in the factories. So they need to characterize, like if you want Miss Universe, not just only female Homo sapiens can be, or Miss Universe, they need to pass a lot of criteria to be one of Homo sapiens and be crowned as Miss Universe. So that to what extent, what do we know about the probiotic? So right now we know that the probiotics, yeah, can reduce the AMR, biofilm, and pathogens, can kill the pathogens directly. They have to act like their own specificities in terms of the host specificity, strain specificities, and the property should be under control of the GLAS or FDA. And this should be free from antimicrobial resistant gene and can be well colonized in gut. Then this is the indications of the probiotics in dog. So you can see the dog with the chronic enteropathies or some of the acute diarrhea or even IBDs or Helicobacter gastritis. The probiotics produced from yeast or some of the singles or multiple strain of the probiotics can be very helpful to reduce this kind of the disordering GI sign. And some of the papers also claim about the urogenital infection and parasitic infestation, preventions of allergies. So what happening? So you eat probiotic and probiotic colonize in your gut, and this can contribute to reduce allergy. So of course you can link these phenomena to the previous one, that the beneficial bacteria can induce proliferations of the T cell called regulatory T cell. And probiotics can reduce obesity because some of the bacteria can produce a lot of energy. And if the bacteria can replace it by probiotics, we can reduce some of the leftovers energy in your GI tract. Once the veterinary want to use the probiotics, what we can recommend? So we recommend to use ahead of weanings and traveling. So if you want to bring your dog or your cat by airplanes, 12 hours travel journeys, so it's better to feed your cat or your dog before journeys. I think 1 week before. And use quite long-term administration. I think it's over a week to a month is a proper preferables. But for chronic enteropathies, we recommend to use several months of application. So in terms of the antimicrobial resistant, yep, right now people are afraid of the antimicrobial resistant gene that may be contained in the probiotic, and some of the product didn't say in the labels of the antimicrobial resistant. So if they recommend to resistant to some antibiotics, so of course we can use currently together with antibiotic, that one. But if the label lack of the antimicrobial resistant profile, then we don't know what's going on inside. So it's better to avoid to use probiotics together with antibiotics. Or if you want to use at least 4 hours apart from each application, would be recommended. Or some of the probiotics recommend, and okay, your probiotics will resistant to amoxicillin. So of course we're not going to use that probiotic with amoxicillin, or even at least 4 hours ahead of use of the antibiotic. This is a recommendation. So one of a very interesting paper that they recommend the use of the probiotic in dog. They divided in 3 populations in the puppies, teenagers, and senile dog. And they observe the parameters like serum IgG, the inflammatory cytokines, and IgA, and the pro-inflammatory cytokine, TNF alpha. So what we expect to see, so once the probiotics were applied, so we need to see the increasing of the IgG, increasing of the interferons, and increasing of IgA, but decreasing of TNF alpha by feeding of the lactobacillus and bifidobacteriums. Then let's see what's going on. Is look, not that simple. So from the puppy dog, the microbiotas that can influence this parameter that I said, you can see the arrowhead directions will represent the correlations between microbiome, between bacteria and the parameters. If they represent a big square, that means very high in influence. And if have their own green, the green one is the positive effect. So look overall of this slide. We have angels and devil. Devil mean the bad bacteria, and this is the good bacteria, lactobacillus. So at the beginnings, the puppies will contain a lot of lactic acid bacteria or good bacteria and have a little bit a devil or bad bacteria inside GI tract, and they can move balancing and outcome to life condition in the growth rate, very well feed intake and very well respond and have very good regulations of immunities. But the time passed to teenage and senile. You can observe the number of devil were increasing until the senile period. So that means that represent the more old, the older the dangers by microbiome member. So to feed the probiotic, the probiotics can convert this kind of the communities, microbial communities, to be younger, to be like teenage. We call this one is the gut rejuvenate phenomenons. Rejuvenate, anti-aging. So become an anti-aging protocol for your gut. Moreover, some of the papers that recommend to use of the probiotics for treatment of the allergies. Yeah, that as the mechanism I mentioned before, the probiotics can differentiate the naive cell to be regulatory T cells. This kind of the cell is very important to down-regulate the activities of the T helper 2. That's why the T helper 2 cannot differentiate the plasma cell and cannot produce IgE. Then if lack of IgE, the IgE will not pin to the mast cells, and the cytokine like histamine, like prostaglandin or leukotriene will not release it at the infective site. So that's why the probiotic can maintain the gut integrities or mucosal integrities for long term. Then in terms of the IBD or inflammatory bowel disease, the use of the probiotics can convert some of the T cell we call CD3 positive, and can differentiate not much naive cell to be FOXP3 or CD4 positive regulatory T cells that can regulate the overgrowth of the T helper 2 and T helper 1, and they can control and they can convert the diversities of the gut microbiome to reducing of the pathobionts and enhancing of the symbiont. Then the dogs will get in normal conditions for a month or half year treatment. So in terms of the viral inhibitory theories, under applications of probiotics, there is a lot of the study, even my study. So I use the probiotics to against PEDs, the parasite epidemic diarrhea that's caused by coronavirus. That this is our result. We can see the low replications and the probiotics can interfere. Sorry, Tarit, what's going on?
Actually, John, I was increasing the sound of the volume,
Okay.
so just mistakenly hit it. Sorry.
Okay. No worry. Let's continue. We success and be published in Probiotics and antimicrobial peptide here, so we can reveal the abilities of our probiotics to against PEDs by interference of the virus attached to the cell. And this one will be can be explained like in terms of the probiotic use in cats, in terms of the FIV. This one also can be interrupt in the gastrointestinal epithelium barriers for FIV as well. And use of the probiotic in dogs, this can shortening the recovery time of the canine parvovirus. And also in humans, they can use for reductions of the inflammations by rhinovirus. The applications in humans can be used for COVID-19 as well. So for using of the probiotics can be another options for prophylaxis of COVID-19 by they said the emerging studies suggest that the capacity of the probiotics to regulate immune response in the respiratory system. Then they can control the inflammatory response. Then the probiotics can work well for improving the development of the small intestine, improving of the growth performance, reductions of mortalities, lowering of the evidence of diarrhea, and enhancing the hemoimmunities, IgG and IgA, and the preventing of the intestinal inflammations or modulations of gut microbiomes or enhancing of the metabolic capacities, or all of the indications is belongs to use of the probiotics. But anyways, in the market, some of the product is not that high qualities. Once we grow in the lab, so the bacteria cannot grow well at 10 to 9 and 10 to 10 as recommended, as the recommendations at the label. So people concerned about that one. But anyways, one of the theories try to explain once you eat the kill probiotic inside, what's going on, and is it still have some this kind of the benefit as the living cell. Of course, I would say most of them still have effective and have some benefit to like this one, except that the modulations of the microbiota, because it's already dying, and enhancing of the metabolic, because the metabolic capacities have to secreted or originated from live cell. So that's why the indications of the some of the probiotic become like just only this one, this sixth one. So that is probiotics. So let's move to postbiotics just a little bit. So I would like to talk about the products from pre and probiotics that can work together and become postbiotics. And postbiotic mostly created in colon space. Look at this one. So from feed and can supply colonizations of the probiotics. And this one, the probiotics can secrete some of the energy chemicals, can make imbalancings, and these the metabolic products can releases and were uptake to the cell and can switch on some of the activities under mucosa. What is it? Yeah, is it secretion of the short chain fatty acids, secretion of the vitamin K, biotin, and folate, and upregulation and absorption of some of the minerals at the large intestine. This is the metabolic functions that relied on postbiotic. Look at the mechanisms. So you don't have to remember this mechanism, but just to understand, at the large intestine, the dominant mechanism is fermentation. The leftover non-digestible carbohydrate will move to large intestine and become fermented under this pathway. And this pathway, you familiar with pyruvate, acetyl-CoA, right? And the byproducts of the pyruvate is lactate, and this is will be converted as the acetate, and the final products of the mechanism will be butyrate. Then what we're thinking about, it's better if we can enhance the productions of this kind of the short chain fatty acids, because this is the sort of the communications compound. Then scientists bring pectin and hemicellulose, is the soluble and non-soluble carbohydrate, into feed, and this can promote because the polysaccharide producers, then the products from this one can produce the short chain fatty acids, especially the pectin and hemicellulose can enhance the growth of Firmicutes phylums, especially the order of Crocididale. And what's going on after they can build a lot of short chain fatty acids? Then the mucosa synthesis and type junction is very strong. This brings to gut integrity and outcome to the healthy condition. And then some of the pathway can get left over of this proteolytic bacteria, and produces that not that balance of the short chain fatty acid will convert the phenomena to be the opposite side. Then I have talking about the compound, metabolic compound that can affect to some other cell, not just only colonic site, but also target of the short chain fatty acids go to brain cells, livers, muscles, and blood vessels. And the target cell is adipose cell, macrophage, neutrophil, T cell. So let's see, this is not just only the intestinal cells. And we need the benefits from this communication is to reduce some of the disease and to enhance the giving some of the energy of the cell, of the GI tract cell itself. So this is the pathway along from mouth to fecal content. So from feed intake, yeah, of course, we need the probiotics with acid tolerance and bile tolerance, and probiotics need to colonize wells along from duodenums and have some effects until the proximal colon. And what we expected from prebiotic here, the prebiotics will come with nutrient, and this is have both soluble and non-soluble prebiotics fiber. This one can help probiotics to colonize, and the microbial digestions will work very well at between duodenum, jejunum, and ileum. and the digestions of the host happenings along here together with microbial digestion. Then go to large intestine. The large intestine, we focusing on aerobic and aerobic probiotic that can create the short chain fatty acid here, beginnings from cecum, and this can promote the anaerobics, especially the group of bifidobacteriums. And the enhancing of bifidobacterium and some of lactobacillus here, yeah, they can colonize well, and they can give short chain fatty acids to be the source of the colonocyte energy. Then the colonic cells get energy, they can proliferate very well. Once they proliferate very well, they can induce the blood supply in the site of colonization. So what happening if the blood supply come here? Of course, the water and minerals will be reabsorbed very well here. Then the waste products from the non-soluble probiotics will be the very important source of the anti-inflammations and antioxidation here. And then they can improve gut integrities. Finally, non-absorbable prebiotics that left until the large intestine can be improve the stool score or stool parameters and can reduce blood glucose here. This is a long way of the nutrients and triggers the miracle of life. Comprising 3 steps. Firstly, energies for colonocytes by producing butyrate, acetate, and propionate. Secondly, they can reduce toxicities by byproducts of the protein, especially indo and branches chain fatty acid and branches chain of amino acids, and production of the phenols. Then the postbiotics, once they released, they act like anti-inflammations and antioxidants, especially the compounds belong to polyphenols. This is the objective key result of your life. So in terms of the safety, the most of the probiotic is very safety bacteria under regulations of the FDA. There is no paper or report about the septicemia from probiotic. That's why it's safe enough. But anyway, sometimes veterinary need to consider about the discomfortable first dose of the administrations of probiotics into pet. So pet may be show constipation or loose stool or some discomfort from bloating gas in the stomach, but it's not that much serious. So you can split or decreasing the dose for pet, like ladder dose. The qualities is depend on stability of the product, the shipment storage. The qualities will rely on the target species and some of the people you can request from the company, the probiotic strain, and the number of the probiotic strain that recommended at the labels of the product. And of course, if you don't know all of that thing, but you know the reputations of manufacturers that can be trustable. So today, we have talked about the miracle of life that's based on prebiotics, probiotic, and postbiotic, and that have their own indications very clearly to the promising application, especially the disorder of the GI tracts, and they can improve the health qualities and stool score, and produces the probiotic as the anti-inflammations and antioxidants. Yeah, of course, some of the things we not see today, but we eat it every day, is become yourself. So something, if you didn't see it, doesn't mean it's not important. Oxygen as well, you breathing oxygen every day, then you healthy. You didn't see any oxygen around your life, your body, right? But you can feel it. You can get fresh from it. So nutrient as well, probiotic as well. Even you don't see what's happening in your bodies, so it can give a miracle of your life. Any question? Are you all still there?
Yes, Ajahn.
Okay, one thing is a miracle of class. I have no homework anymore. So just get into understanding of this new part of your life. If I will be very happy if you can adapt to your life, or you can explain to some of your friends about how importance of nutrient and probiotic, and you can choose the best one for your life. So anyway, if you have no questions, I'm gonna close this class right now.
Okay thank you so much ครับอาจารย์
ขอบคุณค่ะ
Thank you very much, Ajahn.
thank you อาจารย์
Okay, bye.
have a nice day อาจารย์